Thank you very much. Yeah, it's a very exciting time for somebody who is developing technology. Today we want to share some views on how to reconstruct lineage in a cell system, and in particular how we would do this in human systems. So as I said, it's a very exciting time, and many people think this is an exciting time, so science actually chose the single-cell sequencing and single-cell biology in development as the breakthrough of last year. So this is really, really great. And basically one of the major experiments that was highlighted in this breakthrough is to reconstruct the lineage. So if you start with an embryo, and this embryo, during the lifetime of the embryo, will turn into an adult fish. If you now look at the adult fish, can you learn something about what happened during its lifetime? Not by making a video of this fish, but really looking at the endpoint only. And this is actually possible now, basically by encoding history into this fish as it develops. I will show you a little bit more details how that was done. But basically what you do is you were able now to go back in time actually to turn this arrow around and reconstruct what happened during the life of this animal. And of course this is great for development, but what we really want to do is to do this on human tissue, for example on a tumor, so that you have in the clinic the tumor, but then can you somehow reconstruct what happened during the life of that patient, during the life of that tumor, and in this way really understand what went wrong during that process. So that's really the goal of Lifetime, and really the hope that this would work. And what's really the hope that I have personally is that in 10 years we have a cover of Science that looks like this, that the technology that was developed by Lifetime actually is able now to infer histories of tumors, and of course by doing that understanding really what went wrong and hopefully also really cure these diseases. So let me say a little bit more about how people did this during the last year. So there was actually a lot of groups that worked on this together. So it was done on zebrafish, but also in mouse embryos. And last year, as I said, was a lot of interest in this. But basically, this is based on Cas9. Cas9 is a protein, a very interesting protein, that can cut DNA at a very well-defined position based on a guide RNA that you provide. So what these groups did is that they injected purified Cas9 protein into an embryo at the one-cell stage, then also provided the guide RNA. The guide RNA guides the Cas9 to a position in the genome that it can cut. And in many cases, what they choose is basically a transgene, so a gene that, like a fluorescent protein, that doesn't have any function for the animal. And what happens is after you introduce a cut in this particular locus, is that the cell, of course, immediately wants to repair this, because it's not good to have a chromosome broken. So you repair it, but what happens is that this repair is not perfect. It will actually make mistakes. So we call this scars. So after you make a break, it will be repaired, but you remain with a scar. Now that scar is in the genome, which means that it will be inherited by all the future progeny, and therefore you can use this as a kind of a barcode. And if you use many of these GFP copies, you can make a really nice barcode that basically accumulates information during the development. And later on, just by looking at the endpoint, by looking at the adult fish, you can figure out what happened during development. You can figure out which cell is a sister of which cell, which is the grandmother cell, what are the nephews and the nieces. So the whole lineage can be laid out by doing this type of experiment. But as I said, this is done in model organisms like zebrafish and the mouse. Ultimately, we want to do this in human tissue. So we have to think, all of us, about ways to do similar experiments, but then in human tissue. So that's what I want to kind of brainstorm about with you. And as I said, we heard it before, this is really a way to really accumulate ideas today and tomorrow from the community of how to do this. I'm going to give you a few examples how we think this might be possible. So what would replace the Cas9 in human tissue is just the natural process of introducing mutations into the genome. So these are somatic mutations. Every time a cell copies its DNA, it will make a handful of mutations, just makes mistakes. Not many, it's a handful. In a genome with 10 to the 9 bases, there's a few needles in this haystack. So they're tough to find, but this is an ideal marker to look at lineage history. The other thing is copy number variation. This is mostly relevant for cancer, where a lot of the chromosomes actually are copied one time too much or one time too less, and you will have variation in the number of copy numbers across the chromosomes. So this is also something that we're thinking about. A third one, which is actually more unclear, and I would love to get your opinion on this, is epigenetic patterns, so DNA methylation or histone modifications. Many of these marks are also inherited. There are particular enzymes that will copy the epigenetic state from one generation to the next. So this is another thing that we're thinking about. We don't really have any strong, very concrete plans there, but I think it's another option. But I'm going to focus on those first two, the somatic mutations and the copy number variation. So I'm going to show you an example on organoids, and we already heard from Priska a lot about the organoids. It's a similar system, but then it's tailored for looking at cancerous outgrowth. So what happens normally in colon cancer, for example, is that you have a normal stem cell. This stem cell is hit by a mutation, which is an oncogenic mutation, and unfortunately a few more mutations happen in that cell, and this cell becomes basically a cancer cell. It starts to grow faster than the surrounding tissue and overtakes kind of the normal niche, and will start to become more and more heterogeneous. All these different colors are basically different genetic clones, and at a certain point there will be a metastasis that will arise from a cell that escapes from the primary tumor. In an ideal experiment, what you want to do is you want to basically sample this whole process. Say every week you want to take samples, and you want to look at what happens over time. But this is, of course, very difficult in the clinic. But for now, we're going to do this in organoids where this is possible. But ultimately, what we want to learn is how to, when you look at the end point, or hopefully an earlier point here, can you go back in time like we did in the zebrafish by basically looking at the accumulated record of mutations and copy numbers. So the system we use is our organoid. So this organoid system has been developed by Hans Clevers in our institute, where he starts with human wild-type colon organoids, but then he, in the lab, basically puts in the 4 oncogenic drivers for colon cancer. So this is done, in vitro, but then you have a nice system that it's really kind of clear where the mutations are. And what we did is on top of these mutations, we put also viral barcodes in there, so lentiviral barcodes, where basically each cell gets a unique code. So this is a very diverse population of viruses, and they put basically a barcode into the genome of every cell, and this barcode is different for each cell. So what you can do now is you can start to do a kind of an evolution experiment. You start with this culture of organoid cells that are having the potential to form a tumor, and you let them compete amongst each other. And they will, of course, also accumulate mutations, they will change the copy number, etc. So this is 3 different replicas of experiments. So what you see on the x-axis is time here. These are really generations of cells. So we go up to 100 generations. So this is actually a very long experiment. This is about 4 months of data. And what you see in each color is the lentiviral barcode. So you start the experiment very heterogeneous culture with 20,000 different lentiviral barcodes. They all start to compete with each other, some die off, and after 100 generations you're kind of left with a handful of clones that survived. So this, what you see here, is Darwinian evolution in a test tube of colon organoid cells trying to compete with each other, and in the end you have a couple of winners coming out. So it'll be very interesting to see what are special about these winners. So what we now can do, of course, because now we have full access to this system, is we can sample every week single cells from this culture and do our single cell sequencing analysis. And this is the first type of analysis we can do, which is copy number variation. So what you can see now is here are the different chromosomes in single cells. So this is an example of a single cell. So typically it's gray, which means it's diploid. So this is what you would expect in a normal cell, but sometimes it loses a chromosome. So for example, the chromosome 1 here, a piece of chromosome 1 is lost. Sometimes it loses chromosome 18 or 8, but you can already see there are groups of cells. So there's particular clones here that look very much like, they're very similar, so they're probably family members. So this works quite well. You can do this for thousands of cells, look at the copy number, and then group them together in groups of similar copy number states. So it's pretty nice. You can see this at the single cell level, that just this part of the chromosome is just half the concentration of the other ones. Now, what you of course can see in a bulk experiment, if you analyze this bulk, is that you probably would see the loss of chromosome 4 or the loss of chromosome 18, because most of the cells are losing it. But what you never can measure, and that's why it's important to do it in single cells, is that you can say, well, this cell actually lost chromosome 18, but not 4, whereas this cell lost 4 and 18. So this is something you can never get out of a bulk experiment. So another thing, and this is really kind of 10 times more difficult than a copy number analysis, and we're very much still learning how to do this, is to look at somatic mutations. So I mentioned before that every cell division you have 5 mutations that happen in the genome, but you have 3 times 10 to the 9th of them to look through, right? So it's a huge search problem. So we have ways to do this. So first, basically, we look very broadly, and then we focus on particular mutations. But what you see here, an example of about 700 single cells, where I'm showing you here about 10 different somatic sites that are mutated in these particular cells. And what's really important, we can measure 3 things. So one thing which is we're not very happy with is that we don't measure it. So that's unfortunately what most of the times happens if we just don't see it in the single cell, because it's, we start with a single genome and we want to go to a single nucleotide. Sometimes it's stuck to the plastic, or you never see it back. But luckily, there's many cells where we can actually measure it, which will be in red. But also very important is to measure cells that don't have that mutated. So that's in blue. So we basically can measure the presence or the absence of the mutation. And then you can start already to see clustering patterns, but this is still very much in flux, and we're improving this method as we speak. So how to now integrate it? So we basically have 3 pieces of information now. We have a lentiviral barcode, which tells us that the cell was introduced with this code at the beginning of the experiment. Then we have copy number variation that happens during the experiment, and also the somatic mutations that happen during the experiment. And all 3 measurements are done on the same cell. So this is a very nice multi-omics example where you do really 3 different experiments in the same cell. But now you have to integrate it. That is quite difficult. Again, if you have any ideas, let me know. But this is one way we do it. As I said, we have 3 different experiments, and these 3 different experiments change as a function of time. So I can, at the moment, not show you 3 things at the same time, so I'm going to show you only 2 things at the same time. So what I'm showing you here is the copy number tree. So the tree that you see here, so here's where we start. This is the ancestor that then splits into different branches during the experiment. And these branches are defined based on the copy numbers, the changes in the copy numbers. The width of these bands is basically how abundant these are. So if there's lots of cells, in this particular clone 1, it's a wide clone. So the width of it basically tells you how many cells do this. So this is what comes out of the copy number analysis. And on top of it, you find symbols with different colors, and they indicate the lentiviral barcode. So this is the barcode that we introduced at the beginning. Now this experiment already blew us away, because it was, one thing. Yeah, it was quite unexpected for me. I'm also a physicist, so if there's something in biology, I'm always kind of surprised. But I get used to it now. But what you see here is this clone 1, right? It's a very well-defined clone. It actually loses a copy of chromosome 8. But if you look at the diversity in that clone of the lentiviral barcodes, it's amazing. There are probably 20 different barcodes. So this means that there was at least 20 cells at the beginning of the experiment that ultimately lost his chromosome. I would expect that this kind of behavior you see for number 7 is that you find a clone, and it's all basically clonal. You only find 1 lentiviral barcode, because it's 1 cell that ultimately gave rise to the clone, but it doesn't seem to be this. So there are 2 explanations I can come up with. One is that the heterogeneity was already there at the beginning of the experiment, although we didn't see it, because when we do a bulk experiment, we don't see all these losses of the chromosomes. Or these copy numbers happen during the experiment, and they're very reproducible. They happen all the time in different cells, and they are always selected for. So it's very recurrent. Now we can use these somatic mutations to learn, to basically distinguish between these scenarios, because the somatic mutations happen during the experiment, whereas the lentiviral only happens at the beginning. Right? So now we can use this experiment now. So what I'm showing you now is the colors are the somatic mutations. So there are only 2 colors. Red means that it's mutated. Green means it's not mutated. So this is one particular nucleotide in the genome. Of course, I have many more of these graphs, but just to show you. And what you see here is that this particular culture generated different copy number states. They're green, which means that they have the wild type nucleotide. Then a mutation occurred, which turns these kind of green cells into red cells. But what you can see is that they're both green and red cells in this clone here, which means that the cell that had the mutation also lost, in this case, chromosome 4, but the one without that mutation also lost it. Those are two different cells. They cannot be the same cell. So this means that this event happened twice, at least twice, independently. Another thing which we see is that the chromosome 4 is lost. There's, of course, you have two chromosome 4s, you have two alleles, which I call 4A and 4B, and they're also lost independently. So what we see is that many of these copy number changes are very recurrent. They happen over and over again in these cells, in the same culture. Now, when you look at this kind of clinical data, you can also see this type of behavior. So this is patient data from colon tumors, where you can look at the frequency of loss of chromosome 8 and 4, and we see something very similar. We see loss of chromosome 4 is always accompanied by chromosome 18. So this is quite interesting, that these two always come together, and I think that this is something that almost always happens many, many times in a particular tumor. This is not something that is rare. But we're very much still in the process of trying to understand this. So, yeah, so I think this is an example of the first small step towards going towards lineage reconstruction in human tissue. And of course, this is an organoid system, which is good to start, but ultimately we really want to do this in tumors and metastases, right? So, and I think some of this technology that I showed you here will be actually useful in this context. So thank you very much for your attention. I'm happy to take some questions.
Thank you very much. So the questions. There's one over there.
That's very nice. So is it absolutely clear that it's impossible for recombination to occur in these tumors?
So it's a full loss of a chromosome, right? It's a whole chromosome gone. So would you think that would be okay with recombination? I mean, definitely if you have crossing over events and things like that, I mean, but this is a full loss of the whole chromosome. Yeah, yeah. Yeah, so what I'm saying is that different cells lose the same chromosome independently of each other. Yeah. They have a different clonal origin. I mean, I think recombination we shouldn't should not put it on the list as a mechanism. To be honest, the real molecular mechanisms of how this happens are still unclear. Right? What we see is the endpoint. How these things are lost, what is the reason for it, will be the next step actually to understand.
Hi there. So I want to ask about, do you have a system where you introduce the lentivirus multiple times at multiple time points, so you actually can do the same type of analysis as you did, but compare with?
We don't do that. We only introduce it at one point. You could do it. I think what is challenging there is, of course, during the introduction, you typically have also selection again, because you want to select for the virus. So then it's hard to distinguish perhaps the selection during, basically searching for that virus, or the selection that happens because of the competition. But and of course, the problem also is these cells, if you need different, I'm not a virologist, but you cannot introduce the same type of virus, so you need different, I don't know how they call it, but there are different types of viruses to stack on top of each other. It might be possible, but it would be really nice to have something that happens naturally, definitely if you want to use it later for looking at tumors.
Well, we heard in an early talk or so that you actually can trace the cells, right? So you could do the same thing for a cancer organoid.
Yes. Ultimately, these technologies are all developed with the goal in mind to use this on primary tissue in the future. Yes. And of course, these are 20,000 clones you have to trace. It might be challenging for Priska to do that, but we should discuss this.
I have a question. Do you know that those cells are all diploid? And do you have a model of aneuploidy? Because obviously chromosome gain loss often comes about with cell fusion, or let's say failure to divide, and so then you're diploid or tetraploid and then so forth, particularly in tumors.
Yeah. So perhaps you're suggesting there's more of a duplication event first, and then you basically, that's of course possible. And we analyzed the founder culture in great detail. And that one looks pseudo-diploid. Actually, there's in chromosome 1 there's a piece that's missing that is also in the master culture, but all the other chromosomes are close to diploid. Doesn't mean that there's a very small concentration of cells that are in there that have a duplication already. And the only way to make that less probable is to look at the statistics. But it's hard to really prove that it's not there.
Can I? Okay. I have a statistical physics question, yes. And I think I am in front of the right person.
A long time ago, though.
No, this measurement moving from bulk to single cell allows to have information about single samples. So you can talk about joint distribution, conditional distribution. Yeah. So for example, in this case, the fact that some events occur together in a certain time sequence. Yeah. Can you talk about a purely neutral or null model, or can we think about some causal or method behind? Because, for example, for different tumors, there are typical translocation, typical mutations. So is there anything behind this on the structure of DNA, on the time course, or is it purely randomness?
I think it's too early to really go into this type of details. I mean, definitely the probability distributions are a great way to analyze this. They're challenging to analyze because they're in 3 dimensions, right? So we have three different measurements that we want to basically look in the probability space. So it is possible, and then you can look at all the different combinations of joint probabilities. We're in the process of doing this, so unfortunately I can't answer your question at the moment. But definitely, I mean, this is also why we're doing this, I think, is to also get ideas from statistical physics, and other fields to analyze these problems.
I actually link to this. Have you tried to explore if you use different, inputs, differentiation signals, whether you tend to have the same rearrangements, the same chromosome losses, or,
Yeah, so we mostly focused on this particular genetic background, which has these 4 mutations, which is the most aggressive form. We also have the triple mutant and the double mutant. And they are much more boring in a sense that they are much less variable. So we are very excited by the variability, so they are much more well behaved. So it's clearly driven by these mutations. And yeah, we have to do more experiments to really nail down which one is the most important one. But
so last question here.
So you have used a CNV or single nucleotide to follow your cells, but have you thought looking at a region with a high mutation rate such as microsatellite that could be very useful for tracking these cells?
Very good idea. So, and people are working on this. Eudes Shapiro is actually also in the audience, one of the world experts looking at microsatellite instabilities. It's a tough region to sequence, but it's definitely possible, and Eudes really is the person to do this. We don't have full genome coverage, but we have large parts of the genome, so we should, together with Eudes, look at the microsatellites in these cells and see if they're consistent with what we measure for the somatic mutations and the copy number variation. Yeah.